Dear students, welcome you all for the class. In this chapter, we are going to discuss acids, bases, and some salts. I am in the juncture indicators. Indicators are chemical substances which show different colors or different smell in different chemical substances like acids and bases. So, with the help of these chemical substances, we can identify whether the given substance is an acid or base. Because acids are colorless and most of the bases are also colorless. If given substance is an acid or a base, how can you identify it? We cannot taste it because these are all chemical compounds may cause some damages to our body. So we have to select some other ways to test whether the given substance is an acid or a base. So for this purpose we are using some chemical substances. They are called indicators. So indicators are chemical substances which change their color or smell in different types of substances. This can otherwise defined as indicators are chemical substances which give different color in acidic medium and some other different color in the basic medium. There are three different types of indicators. Indicators can broadly classified into three types. They are natural indicators, synthetic indicators, and olfactory indicators. Natural indicators are indicators. They are the natural substances. They show different color in acid and some other color in the base. So indicators which are obtained from the nature, then they are called natural indicator. Examples for natural indicators: litmus, turmeric, red cabbage. You can see the red cabbage here. Then leaves extract of the flowers like hydrangea, then petunia and geranium plant. The pictures are given here. So these are some of the natural indicators. So these are the indicators. They are obtained from the nature or from natural sources. So they act as indicator. They help us to identify whether the given substance is an acid or a base. So litmus, let us consider litmus. It is a natural indicator. Let us study few important points about litmus. Litmus is a natural indicator. It is obtained from lichens. Lichen are organisms in which algae and fungi live together. They grow on the rock surfaces. So lichen is an example for symbiosis. In biology you are going to study symbiosis, where different types of organism which are mutually benefited can live together. Lichen is one such organism in which algae and fungi they live together and both are benefited in this relationship. So if you get some extract, a solution from the lichen, that is called a litmus solution. This litmus solution is generally purple in color. Its color is purple and it can be obtained in two forms. It is available in two forms. One is it is available in the form of paper, solution, litmus paper, and it can be obtained in another form that is called solution form. So litmus can be available in two forms, that is paper or solution. When this litmus is the two, blue litmus when added to acid. See, blue litmus turn red in the acidic medium. Then red litmus turn to blue color in the basic medium. So based on this action of red and blue litmus on a given substance, we can easily identify whether the given substance is a base or an acid. See, this is a well-known material used for preparing food, that is turmeric. It is also an indicator. Turmeric is generally yellow in color. So, when turmeric solution or when it is applied on the paper, it turns reddish brown with the base. For example, if you take what you call turmeric powder in some vessel, add few drops of lime water, calcium hydroxide, then the color yellow turns to reddish brown color. It indicates that the given substance, lime water, is a base. So it changes from yellow color to reddish brown color. So by changing its color, we can identify whether the given substance is a base or an acid. So turmeric can also be used as an indicator. So now you have the knowledge and you can answer this question. What happens to a stain of curry on a white cloth when soap is scrubbed on it? So soap is a base. Here turmeric is present in the curry. So it is yellow in color. If you scrub it with soap, that means a base, then yellow color turns to reddish brown color. Okay, so next let us go to the next type of indicators. They are synthetic indicators. So synthetic indicators are indicators, they are artificially prepared in the laboratory. So they are man-made indicators. Example, phenolphthalein and methyl orange. These two are the synthetic indicators. They are produced or prepared by man. So they are called synthetic indicators. Among them, the first one is phenolphthalein indicator. It is synthetic and it is colorless indicator. That means phenolphthalein indicator does not contain any color of its own. It remains colorless if added to water. So if you add few drops of phenolphthalein indicator to add as acid, no color change in the acid. Acid is also colorless, phenolphthalein is also colorless. But when phenolphthalein indicator is added to base, it turns to pink color. So the colorless phenolphthalein indicator gives pink color in the base. So with the help of this, we can identify whether the given substance is an acid or a base. Then another synthetic indicator, that is methyl orange. It is used to show acidic or basic nature of the substance, but this is better. Originally the methyl orange indicator has orange color. This orange color changes to red color in the acidic medium and turns to yellow color in the base. So the same orange color, original color of the methyl orange is orange color. When it is added to acid, it turns to red color, orange to red color. When it turns to orange to yellow color, then the given substance is a base. So that is about synthetic indicator. There are some indicators, they are called olfactory indicator. Olfaction refers to the smell. So these are the substance, they generally have some specific smell of their own. But when these smelling substances added to acid or base, they change their smell. So based on this, we can identify whether the given substance is a base or an acid. So olfactory indicators, example for olfactory indicators, onion, then vanilla, clove, etc. Onion, take onion in the form of paste or juice of onion, then it loses its smell when added to base. If you add base to onion smell, the smell of onion disappears, it vanishes, but it does not change its smell with acid. So if you add acid to onion paste or juice, the smell does not change, it remains as it is. Similarly, vanilla also. Vanilla has a pleasant smell of its own. When vanilla is added to base, its smell vanishes, but it does not vanishes with acid. So based on this, we can identify whether the given substance is an acid or a base. Thank you for listening the class.